Hi, I'm Trey.
And I'm Sam.
And I'm Lainey, and we're all coastal environmental science majors. We're currently at Geelong's Fish Camp, which is also the home of the Matanzas River Keepers, and we are beyond excited to bring you along our Capstone journey. This is the Ocean Palms neighborhood that houses 8 detention ponds that are used to irrigate their lawns. 2 of the 8 are connected to the nearby Matanzas River, resulting in saltwater intrusion. Residents were interested in taking measurements to assess nutrient levels and salinity.
While helping out the neighborhood, we made our own hypothesis as to what we were going to find. There will be a difference in salinity levels between the ponds that are connected to the inlet and those that are not. If the ponds have different salinity levels, there will be a difference in nitrate levels due to salinity. If the ponds have different salinity levels, there will be a difference in phosphate levels due to salinity.
Here I am grabbing duplicate surface grabs from one of the detention ponds. While I perform this task, Trey is using a YSI exosonde to gather water quality parameters of interest behind me.
The first thing we do in the lab is filter our grab samples through a microfiber filter and store the filter and filtered water separately in the filter.
We come back the next day and begin by pipetting the filtered samples into separate groups of vials, where one group is for phosphate and the other nitrate. In between pipetting, we put a small amount of each sample into a bottle to create a matrix. The pond matrix is a representative sample of all the ponds combined.
The fluid I am pipetting into the vials is a nitrate reagent which will allow us to determine the nutrients levels within our samples. The process is then repeated for the phosphate reagent.
I'm putting something we call standards into our vials with the pond water matrix. Standards are a known amount of nitrate or phosphate. I put increasing increments of each standard into the vials so we can compare these known results to our unknown samples.
After we add all of our reagents and standards into the vials, we place them in an oven to incubate for an hour.
To figure out our nitrate and phosphate levels, we have to use this spectrometer. We pipette samples from each vial into a cuvette that holds the liquid inside the machine. We close the lid and run the sample. This machine runs light through our sample to give us units in RFU, which are relative fluorescence units, which we will later convert to micromolar units.
We take the filters from our first step in the lab, filtration, and we add 90% acetone, which will pull the green chlorophyll A pigment off of the filter. To expedite this process, we use sonification. This process uses sound to disrupt cell membranes and release content.
We then pipette the chlorophyll A sample into a cuvette and place them inside the fluorometer, a similar process to the nutrient analysis shown before.
After crunching the numbers and analyzing our data, we are excited to show you our results.